All right. So reinforcement learning in neuroscience has also kind of a bidirectional influence. So reinforcement learning was influenced by neuroscience. For example, eligibility traces are inspired by certain properties of synapses. And now there's also accumulating evidence that the nervous system might implement aspects of reinforcement learning. So again, similar to psychology, sometimes parts of the RL algorithms are inspired by neuroscience, but also neuroscientific experiments are conducted based on reinforcement learning models or mechanisms and their predictions. Okay, so first of all, one important factor we need to introduce is dopamine. So dopamine plays a role in many important processes in the brain, like motivation and action selection, and it's also involved in diseases like, for example, addiction, schizophrenia, and Parkinson. And understanding the exact role of it and the mechanisms might help us also understand these diseases and processes. Dopamine is an important neurotransmitter for reward processing in mammals. But it's also called a neuromodulator because it has so many functions besides just direct excitation and inhibition of target neurons. And triggering dopaminergic neurons is enough reward signal to perform classical and instrumental conditioning in mice. So basically, they did conditioning experiments with mice, but instead of giving them a reward like a drop of juice or something, they actually just stimulated dopaminergic neurons in the brain. And that was enough signal to condition these mice. And there were also similar experiments with fruit flies, but in fruit flies, it seems to be to act as punishment. So it seems like dopamine has a different effect on the mammal nervous system and on nervous system of insects. But in both, dopamine seems to act like similar to TD error in reinforcement learning. So not directly as a reward signal, but more informing learning. And those findings lead to the reward prediction hypothesis. And the idea is that dopamine seems to convey a TD error when learning and decision-making takes place. And the hypothesis is that dopamine-producing neurons deliver an error between old and new estimate of expected future reward through their phasic activity. And that idea was put forward in 1996. And in experiments, they compared the TD error from TD lambda with phasic activity in dopamine neurons during classical conditioning experiments and found some very interesting overlaps. And since the TD error can actually be negative, but the firing rate of a neuron can't be negative, they define the firing rate as the difference from the neuron's average background firing rate. So is it higher or lower than the average firing rate of the neuron? And for their TD model, they use a complete serial compound representation. I hope you remember from earlier. Okay, and the TD error and the dopamine neurons will show similar effect, particularly in both of them, a response only occurs if a rewarding event is unpredicted. The neutral cues that precede a reward don't cause a strong response, but after learning, the cues gain predictive value and start to elicit responses. So similar to the classical conditioning in the dog, but no on a neuronal level. If you show a neutral stimulus, there's in the beginning not much response, but once it is associated and learned, then the neutral stimulus starts to have predictive value and also cause responses in these dopaminergic neurons. And if an earlier cue reliably precedes a cue that has already been learned, the response shifts to the earlier cue and ceases for the later one, which we also saw in these classical conditioning experiments on a behavioral level. And now they find this on a neuronal level as well in the firing of the dopamine neurons. And last thing, if the predicted reward is omitted, the neuron's response decreases below its baseline, so negative prediction error, shortly after the expected time of the rewarding event. Okay, let's look at some experimental evidence for this. So Romo and Schulz in 1990, they recorded dopamine neuron activity in monkeys. And well, in general, we already know that, or knew back then, that dopamine neurons show bursts of activity in response to intense, novel, or unexpected visual or auditory stimuli that trigger movements, but they are not directly related to the movements, but rather to the novelty or unexpectedness of the stimuli themselves. And in their experiments with monkeys, they had monkeys reach into a bin with food in it. Food is not visible from the outside, and they observed a phasic response in their dopaminergic neurons when the food was actually touched. If the bin was empty or there was something else in it, like just a wire, then there was no response in the neurons. After training the monkey with the setup, so reaching into the bin and finding food many times, the dopamine neurons actually don't respond to the touch of the food anymore, but they respond to the sight or the sound of the opening food bin. So the earlier predictive stimulus predicting that they will soon be able to reach the food. So the phasic response has shifted from the reward to the stimulus preceding the reward. And it looks like the dopamine neurons correspond to TD errors and not just the simple Rascona-Wagner errors. And that's what they investigated in a series of experiments also in Schulz's lab 2 years later. So they trained monkeys to get juice by pressing a lever after a light is illuminated. So once the light turns on, they can press the lever and then they receive some juice. Apple juice, I think. And so in the beginning, let's see. In the beginning, we have here the response after the juice is received, an unexpected delivery of apple juice or a reward. And then after training, the lever pressing is, or after training, the dopamine response happens in response to the light instead of the juice. So now the response is actually shifted forward to the predicting aspect of the reward. And here, when the juice is actually delivered, there's no spike in the neurons anymore. And after more training, the lever pressing gets faster, but the number of dopamine neurons that respond are getting lower because it's not that novel anymore or unexpected. And then they performed a second experiment in which they had actually 2 levers with a light above each of the levers, and the light above the levers serve as a cue about which lever should be pressed at the trigger cue. So first, one of the 2 lights turns on, then they have to wait until the trigger cue, and once the trigger cue appears, they are supposed to press the lever under the light that had turned on earlier. So the light is the instruction, then we have the trigger when it can press the trigger, and then it receives the juice. And now the dopamine response actually shifts even earlier here to this instruction cue and fades away here at the trigger or at the reward side. What was interesting, what they observed additionally, is that if the monkey pressed the wrong lever, the dopamine neurons show a sharp decrease after the time when the reward would usually be received. Okay, so here, if we have no prediction and the reward occurs, so that's before any kind of learning happened, we get a TD error here after the unexpected reward was delivered. Then after learning, we get a TD error here when the trigger stimulus comes, which predicts the coming reward, but nothing changes here where the reward is actually delivered. And then here, this is also learning. It also gets a TD error here, predicting that soon the reward will appear, but now it doesn't receive the reward. So it actually has a negative prediction error at this point. So it expected a reward, it didn't show up, so the prediction error is actually negative here in these dopaminergic neurons. And this is consistent with the behavior of the TD error in TD learning. So here, this is now in the TD learning model. If we're here early in learning, we have no value estimate, and we have a TD error here when the reward is actually delivered because it's unexpected. Then learning is complete. We have a high value estimate from this onset on from the trigger stimulus. And the trigger stimulus, we don't know when it actually comes, so there's still some error when it starts. And then when the reward is submitted, we actually still have the high value estimate, we still have the error in the beginning, but then we have the negative TD error at the point where we predicted to receive a reward. Okay, so here we have a match between the model and the findings that they find in the dopaminergic neurons. Okay, but there's a difference between the TD error, at least when we use the complete serial compound representation, and the dopamine activity if the reward is delivered earlier than expected. So in the TD model, it first gets a positive TD error when the earlier unexpected reward is delivered, but then still has a negative TD error at the time when the reward was expected to be delivered. But the dopamine neuron activity doesn't drop below the baseline at the usual reward time if the reward has already been delivered earlier. So it's like, okay, I already got my reward. I got it earlier than expected, but I don't expect to get another reward now at this timing. So there's a little difference between the model and the actual experimental findings. And so it seems like there's something more complicated than just TD learning going on, at least with the CSC representation. And if we use the microstimuli representation, so if you remember, that was the one with the smoother curves, that fits the dopamine activity a bit better. And also some variations of the CSC representations, like prolonged eligibility traces, also improve the fit a bit. And as an alternative to explain this, Daw et al. In 2006 proposed that the brain's TD system actually uses representations produced by statistical modeling carried out in the sensory cortex instead of representations based on raw sensory input. And that could also explain this discrepancy. Are there any questions so far? Okay, then I would go on to the neural actor-critic. So there's this hypothesis that the brain might actually be implementing an actor-critic-like algorithm in which the TD error plays an important role. So the idea is that, so in general, in actor-critics, Leon talked about it shortly yesterday, the idea is that the actor learns the policy and then the critic criticizes the actor's actions. So there's kind of like a adversary thing going on between the two. And actor and critic, the idea is that they could be realized in two parts of the striatum, so the dorsal and the ventral striatum respectively. They could both use the TD error as a reinforcement signal, but with the TD error having different effects on the two. And there are similar observations in the brain which kind of support this idea. So there are axons from the dopamine neurons that target dorsal and ventral parts of the striatum, but the way in which the dopamine influences the synaptic plasticity in those two parts depends on the properties of the target structure. So the dopamine doesn't have to be like a master reward signal, but it could rather be some reward-related information like a TD error, which has different effects depending on if it goes to the dorsal or ventral striatum. And the idea is that it's like similar to the actor and critic, where the TD error also has different effects on the actor and on the critic. And here's like a really rough comparison of the two, or like the idea how they could relate to each other. So here, first of all, on the left side, we have how the actor-critic is implemented in reinforcement learning. So we have here the TD error, which is then sent, so it criticizes basically the actions of the actor. So actor and critic both get the same features from the environment, and the critic produces a value estimate, which can then be used to calculate the TD error from the reward of the environment. And this TD error can then be used to criticize the actions that the actor selected here. And then the actions get sent back to the environment. And in the brain, how it is suggested is now shown on the right side. The idea is that the TD error could be transmitted by dopamine neurons in the ventral tegmental area and the substantia nigra pars compacta to modulate changes in the synaptic efficacies of the input from cortical areas to the ventral and dorsal striatum. And the idea is that the actor tries to keep the TD error as positive as possible, and the critic tries to keep the TD error close to zero, so making close to optimal value predictions. Here. Okay, so in the actor, the TD error would inform how to update the action probabilities to reach higher valued states, so keeping the TD error positive. And in the critic, the TD error informs about the direction and magnitude of the change that is needed at the parameters of the value function to improve its predictive accuracy. So trying to keep the TD error close to zero, having as close as possible match between the value estimate and the received rewards. Some findings in the brain basically suggest that this might be realized with the dopaminergic neurons in the striatum, and having the critic in the ventral striatum and the actor in the dorsal striatum. Okay, that's the suggestion. Is that clear so far? Okay, and the difference between the two, so between the actor and critic, is in the learning rule, in the way that the eligibility traces are used. So here, maybe I can get both. Okay, so here are the two learning rules for the two. So in just the way the eligibility traces are updated. So the actor uses contingent eligibility traces. What does this mean? It means it uses a 3-factor learning rule. So for the update, it needs the presynaptic activity, the postsynaptic activity, and the eligibility trace. So 3 factors to make its update, which means it takes into account which action was actually taken, so the postsynaptic activity. And this is different in the critic update rule, because the critic only uses a 2-factor learning rule, which is just the presynaptic activity and the eligibility trace, so it doesn't take into account the postsynaptic activity. For the actor, it's crucial to also take into account the postsynaptic activity in order to keep the information about the actions that were taken, which is needed to assign credit or blame among the different policy parameters. Okay. And a more realistic model should actually also take into account the timing, because it takes several tens of milliseconds between the arrival of a presynaptic input and the postsynaptic firing of the neuron or inhibition. And this is especially important for more realistic actor units. So because in the actor unit, we actually use the pre and postsynaptic activity. And there's this concept of spike time dependent plasticity, so STDP, in the brain, which is a type of heavy plasticity which might be how the actor-like plasticity is implemented in the brain. At least that's the idea. And so basically the idea is that changes in the synapses efficacy depend on the timing between the pre- and postsynaptic action potential. So in the most studied version of STDP, the synapse increases in strength if the incoming spike arrives shortly before the postsynaptic neuron fires, and decreases if it arrives shortly after firing. So it's kind of like, if it arrives shortly before, it seems to have contributed really to the firing, so the strength should increase. If it arrives shortly after, it shouldn't contribute to increase of strength, because it couldn't have contributed actually to the firing. And there's also the reward modulated STDP, which is like the three-factor form of STDP. So besides the timing of pre- and postsynaptic activity, we also look at the arrival of some neuromodulatory input like a reward or dopamine, which must follow the appropriately timed post and pre- and postsynaptic spikes. And there's some evidence that the reward modulating STDP occurs at the spines of the medium spiny neurons in the dorsal striatum, where we think that the actor is, with dopamine as the neuromodulatory factor. So that would be kind of like the actor learning rule that we think might be happening there. That could be explained with the reward modulated STDP, or implemented with it. Okay, then we have this idea of hedonistic neurons, which was proposed by Klopf, 1972, where the idea is that individual neurons just want to maximize the difference between the synaptic input that is treated as rewarding and the input that is treated as punishing, by adjusting the efficacy of their synapses. And this can be trained with response contingent reinforcement, so the three-factor rule, using contingent eligibility traces. So the one that is used for the actor, with looking at the pre- and the postsynaptic activity and the eligibility trace. Okay, so Klopf proposed here the Klopf traces, which are like a bit more complex version of the exponentially decaying eligibility traces that we used in TD lambda. And these traces, he proposes, reflect the duration of many feedback loops that the neuron is involved in. So each neuron can be involved in like a lot of different feedback loops, and these feedback loops can be inside the brain or the body, or extend out into the world through the motor or sensory system. And the shape of these traces, so each synapse has a trace associated to it, and the trace of a neuron is like a histogram of the durations of all the feedback loops that it is involved in, with the peak at the duration of the most prevalent feedback loops. And this type of traces would is more complex, but it would actually have some computational advantages in complex reinforcement learning systems by refining the credit assignment problem or process. And also Klopf, this idea that these neurons just want to maximize that difference is that, yeah, if you bring it to the extreme, he basically says that many aspects of intelligent behavior can be understood as just the result of collective behavior of a population of self-interested hedonistic neurons. So each neuron just following kind of its own goal, but to have the best outcome, they kind of need to cooperate. And this is what the field of collective reinforcement learning studies, which is the study of the behavior of populations of reinforcement learning agents. And it is relevant for social and economic systems. But if Klopf is right, that neurons might actually be just like self-interested hedonistic neurons working together, like every neuron being its own agent, then it might be relevant for neuroscience as well. Okay, so if we look at the learning in a population of actor units, which all want to maximize the same reward signal, so the TD error, we can look at this as a cooperative game or a team problem. So we say in the actor, each actor unit is its own agent, and they all receive the same global reward signal, but they need to do different things in order to have like a cooperative action and together receive the highest possible reward. If they're, so in this case, there's no conflict of interest. They all want to maximize the TD error. So this is a cooperative game. And yeah, they have a common goal that needs to be reached through cooperation. Okay, in this case, in this case, again, we need to address the credit assignment problem because we now have a team of actors. And how do we figure out which team members actually deserve credit or blame for receiving the reward? And to solve this problem, we can use the contingent eligibility traces, which is one part to solve it. It's not enough on its own, but it's definitely necessary, because if we use non-contingent eligibility traces, so the ones that just use the presynaptic input and the eligibility trace and don't look at the postsynaptic output, that wouldn't work in the team setting, because there would be no way to correlate actions with changes in the reward signal. We can use actually non-contingent eligibility traces to predict, but not to control. So we can use them in the critic, where all agents would predict the same quantity, so the value estimate, but then there's no need to produce different patterns. So there's no team problem. So everyone's just predicting the same quantity. There's no need for cooperation in this case. But now in our team problem of control, we do need these contingent eligibility traces. Okay, and additionally, we also need to estimate the statistical correlation between team members' actions and changes in the team's reward signal, and that needs to be estimated over many trials or experiences. Okay. Also, we need to have variability in the collective actions. So we want to explore the space of collective actions, and the simplest way to do this, if we have individual exploration of each agent's individual action space, which will automatically lead to collective exploration of the action space. But there are also more sophisticated approaches. I'll get to that in a moment. Here, and then Williams in 1992, he talked about if we take a team of Bernoulli logistic reinforced agents, which do basically policy gradient learning. These agents, they implement policy gradient algorithm with respect to the average rate of the common reward signal. And then this team can actually ascend the average reward gradient. And if the team members are interconnected, it can be like a multi-layer perceptron. So it basically learns like a multi-layer perceptron, but backpropagation is replaced with a broadcasted reward. And this is a really interesting idea because, well, in practice, backpropagation is a lot faster, but using reinforcement learning with this broadcasted reward over a team of agents is actually biologically a lot more plausible as a neural mechanism. So for example, there's a really wide dispersion of dopamine signals in the brain, which could implement such a mechanism. So I think it's a really interesting approach to learning, or as an alternative to backpropagation, which could actually be implemented in the brain. And if the team members can communicate with each other, the exploration of the collective actions can actually be more sophisticated than just combined individual exploration, so they can coordinate actions and focus on parts of the collective action space. There are also some other mechanisms besides the contingent eligibility traces, like, for example, winner-takes-all type of learning, where the collective actions are decided by one or a few members of the team, and then credit and blame can simply be given to those who won the decision and made the decision. So there we solve the credit assignment problem in the way that just a few or one member makes the decision, and then we know who's to blame or reward. Okay, almost done. Let's talk about a few model-based methods in the brain. So the Actor-Critic is not model-based, it's model-free. And for goal-directed behavior, we could use a model. And this model that estimates, for example, the transition probabilities and rewards, and that we can use for planning. And experimental evidence suggests that placing the actor in the dorsal striatum is actually too simple, and that there seems to be a difference between the dorsolateral striatum and the dorsomedial striatum. And the idea is that they might be responsible for model-free versus model-based processing, respectively. That's based on some evidence. So, for example, if we deactivate the dorsolateral striatum, habit learning is impaired in rats. And if we have rats where we deactivate the dorsomedial striatum, the goal-directed processing is impaired. And there were some studies with humans and primates which also supported these findings. Additional to the dorsal striatum, the orbitofrontal cortex also has some indications to be involved in goal-directed choice. So, for example, there's strong activity related to subjective reward value of stimuli and expected reward as a result of actions. And also the hippocampus seems to play a role in the model-based behavior because it seems to be critical for memory, also spatial navigation, and it seems to be important to imagine new experiences. So plan ahead, imagine the future, imagine possible futures. So for example, when we have a rat that gets to a choice point in a maze where it needs to go left or right or straight, for example, then the space representations in the hippocampus sweeps forward along possible paths, and this corresponds closely to the path that is then taken. So the hippocampus may be actually critical for representing these state transitions in the internal environment model, and to use this model then to simulate possible future states. But in general, there's not enough experimental evidence yet to make any definite claims here, and it's not even really sure if there is a separation between the neural substrates for model-free or model-based learning. So we are not sure yet how it actually works in the brain, but there are some indications about which parts of the brain may be responsible for implementing different aspects that are similar to reinforcement learning aspects, like model-based and model-free learning, actor and critic type of learning, TD errors, and so on. And last but not least, I just want to make a little extra point, which is why I think this topic is even more interesting, is because, okay, it's not just to play games and solve optimal control problems, but you can actually try to understand learning and behavior and psychological diseases even, which I think is like super interesting to have these types of models to try and explain things that might be going wrong, and actually use these models to find better treatments for these things. So, for example, we have addiction, and the idea is it may be caused by certain drugs that exploit our reward-seeking behavior and wiring. And if we can understand our reward-seeking behavior better, we might be able to understand things like addiction better as well, and also to treat it better. And why we think this is that many addictive drugs lead to direct or indirect release of dopamine in the striatum. And Reddish, 2004, proposed that some addictive drugs, like cocaine, for example, produce a temporary increase in dopamine. And this dopamine surge increases the TD error in a way that can't be compensated by changing the value function. So the drug reward can't be predicted away by the brain. And that leads to the problem that the value of these states increases without bound and makes actions that lead to those states preferred over all others. But of course, addiction is very complex, and also not all addictive drugs involve dopamine. And for example, there are changes involved in the brain structure itself with long-term drug use, and also with some drugs you keep needing increased dosages for the same effects and so on. So of course, this is not the whole explanation, but it is an interesting approach to trying and explain it. And this is actually a new and emerging field called computational psychiatry, where they try to understand mental disorders through computational models. So there are some super interesting theories, for example, on schizophrenia, depression, anxiety, addiction, panic disorder, Tourette syndrome, autism, bipolar disorder, OCD, ADHD, personality disorder, eating disorders, and others. And they try to use computational models to understand them better. And many of these models are based on reinforcement learning and the brain's reward system. So I think this is a super interesting direction and application of reinforcement learning models as well. And if you're interested in this, there's the first introductory textbook into this field. It was released just a few months ago in November. And yeah, you can find it online and buy it. Yeah, if you're interested in this field.